Hi, I'm going to talk a little bit about the methods we used to produce essential fish habitat maps for the Mid-Atlantic Council. Um, so most of the quantitative data that we have on habitat use for fish on the Northeast Shelf comes from random sampling conducted by fishery independent surveys, such as the Northeast Fisheries Science Center bottom trawls. However, these surveys really sample just a tiny fraction of the seabed each year, effectively providing snapshots of the underlying true animal distributions at discrete and often far scattered points in space and time. To develop a more holistic picture of what true animal distributions look like, we need to try and fill in the gaps between these sparse data points. Species distribution models, or SDMs, are a commonly used statistical approach that relates observed patterns of abundance, for example, in fishery surveys, to environmental conditions at the time and place that animals are captured. And over many observations, we can draw inferences about species' relationships with the environment that likely reflect their physiological tolerances or their ecological requirements, for example, or their thermal tolerances or their relationships with physical structure. We sometimes call this their habitat niche. And we can then take those inferences, this estimated niche, to predict where a species is likely to occur based on ambient environmental conditions, effectively filling in the gaps between snapshots in a sort of mechanistic way. But species distributions aren't solely determined by the physical environment. They're also affected by the distribution of other living organisms through interactions that occur between them. Competition, predation, or facilitation can produce interdependencies in the arrangement of animals, or correlations in species abundances that aren't explained by the environment. By explicitly accounting for these correlations, so-called joint SDMs, or JSDMs, that model multiple species at once, can obtain more precise estimates of species-environment relationships, while also offering potentially useful ecological insights about the relationships between species. With this in mind, for the EFH models, we employed a spatiotemporal joint SDM that approximates species responses to the environment as smooth functions, like a generalized additive model, or GAM, allowing for realistic, nonlinear, niche responses without imposing strong assumptions on the shape of their relationship. At the same time, the model estimates relationships between different species, or the correlations, through spatial and temporal random effects that are drawn from a species' common distribution, hierarchically, allowing for the borrowing of information across species, which can also help with estimation for rare taxa. Biological observations, or catch data, that inform the model come from 4 different bottom trawl surveys, with the NMFS spring and fall surveys supplemented with nearshore observations from the NEMAP survey, as well as the Massachusetts and Maine, New Hampshire state surveys. It's fitted to 20 years of data comprising about 24,000 distinct observations. And to account for stage-specific differences in habitat use, each species, with the exception of rare ones, is modeled as 2 distinct groups, adults and juveniles or subadults, with individuals assigned to life stages based on length and maturity data from the literature. Environmental covariates that drive the model include time-varying physicochemical predictors like temperature and salinity, as well as static variables that quantify conditions over the longer term, such as the strength of wave or tide-driven currents, or basic aspects of the seabed like sediment grain size or topographic complexity. The model also includes parameters that account for survey-specific differences in catch efficiency and swept area. Environmental predictors applied for a given species are dependent on the water column use of that species, so models for pelagic species are informed with covariates that reflect surface conditions, while those for demersal species reflect conditions near the seabed. The continuously gridded model domain that's used to produce predictions corresponds roughly with the bounds of the bottom trawl surveys and has a 1 kilometer spatial resolution with about 270,000 grid cells. Predictions are made using monthly mean conditions for 3 months each spring and fall for 20 years. In order to quantify uncertainty in the predictions of the model or its sensitivity to the data that's been used to train it, we carry out a tenfold cross-validation procedure that involves refitting and predicting for the model 10 times, each time omitting a distinct 10% of observations from the dataset. And from this, we obtain a coefficient of variation that reflects the variability in predicted counts for each grid cell across all 10 model folds. This also allows us to assess out-of-sample predictive performance across every observation in the dataset. So I thought it might help to look at how this works on a single species. So here we're looking at juvenile goosefish or monkfish with lengths less than 37 centimeters. And the plot on the left depicts the location of all the trawl samples over the 20-year period, with color indicating which of the different surveys it is. And the plot on the right is showing only the positive occurrences of juvenile goosefish, with the colors depicting the observed counts, with warmer colors showing higher counts. So the model was fit to bottom conditions, since goosefish are demersal creatures. And here you can take a look at the estimated niche responses across the 5 most important environmental predictors. So the predictor values for each distinct sample are plotted in color in the top plot, the spatial plots, while the smooths, the response curves that have been estimated, are below them, where the gray band represents a measure of sort of uncertainty around the predicted shape of the smooth there. So the x-axis on these smooths represents the value of the covariate, and the y-axis represents the estimated effect on goosefish abundance. So you can interpret the first smooth as indicating that goosefish abundance begins to decline when bottom temperatures exceed about 15 degrees Celsius. Similarly, abundance is greater at high marine salinities. It's positively related to depth. And declines at locations where tidal current velocities are on the higher end. And it has a convex relationship with the grain size here that indicates a preference for sediments that are more fine-grained sand or muddy bottoms. Then, given the fitted models, we can go back and predict for each of the 10 folds to obtain out-of-sample predicted counts for each observation in the 20-year dataset, so that we can evaluate predictive performance. On the left, you can see the observed data, and on the right are the predicted densities. Then we can use the full model, which is fitted to all the observations, to predict on a continuous 1-kilometer grid for each of 6 months, 3 in the spring, 3 in the fall, across the full 20-year time frame. Finally, we average the counts in each grid cell across the full modeled time period to obtain a 20-year mean predicted density for each cell. Then, after dropping cells with 0 or near 0 rates of encounter, the cells are ranked based on their density and assigned to quartiles. The upper 3 quartiles, which are yellow, green, and blue in the figure on the right, together delineate the EFH boundary. Then, last but not least, the model-derived EFH boundary is then combined with additional inshore boundary information to produce the final EFH footprint.